Before we move on to the path section of the chapter, we have to review just a few more basic concepts relevant to the anatomy and physiology of the musculoskeletal system. One of these topics is the process of muscle contraction and the conduction of electrical signals that lead up to it. These two pictures highlight some of the basic structural components of muscles at a cellular level and a more structural level. In Figure A, you can see the cellular and receptor level detail of individual myocytes. The invagination that's highlighted represents a T-tubule. On the surface of the cells, you have dihydropyridine receptors, which are voltage-gated calcium channels. Within the cell, lining the sarcoplasmic reticulum, you have ryanodine receptors, which are calcium-induced calcium channels. The sarcoplasmic reticulum and associated channels serve as storage areas for calcium and selective passages for calcium flow, respectively. Calcium, as you may remember from your physiology class, is integral to the process of muscle contraction. In B, you can see the ultrastructure down to the subcellular microstructure of a fascicle. You can think of muscle fascicles as made of muscle fibers, which are themselves made of myofibers. Myofibers are made of sarcomeres, which are the smallest functional units of muscle. The different bands, zones, and lines detailed in the picture of the sarcomere are worth looking at. In general, the sarcomere extends from Z line to Z line. You can remember that Z is the end of a sarcomere and also the end of the alphabet. Off the Z line, actin molecules project inward toward the M line. Off of the M line, you'll notice thicker lines that represent myosin that's extending out toward the Z lines. The thin actin fibers and thick myosin fibers overlap to some degree. Of course, they'll overlap more when muscle is contracted and will overlap less when relaxed. The M line is just the midline of the myosin chains. The H band represents the part of the myosin chains that do not overlap with the actin. Similarly, the I band represents the part of the actin chains that do not overlap with myosin. However, if you think about it, these two areas will get smaller with contraction as the area of overlap between actin and myosin increases. Finally, the A band is simply the length of the entire myosin chain from end to end. So let's back up a second and review. The H zone contains myosin only, the I band contains actin only, and the A band contains myosin and the myosin-actin overlap region. The Z line is the site of actin attachment, and the M line is the site of myosin attachment. To remember the distinction, you can use the memory aid His shrinkage, H-I-Z, and the A band is always the same. If that didn't make sense already, we'll review these concepts as we work through the steps of muscle contraction. The first thing that begins all muscle contraction is an action potential depolarization, which travels down the neuron and opens voltage-gated calcium channels presynaptically. This causes neurotransmitter release. Once the neurotransmitter reaches the postsynaptic neuron, the muscle cells begin depolarizing at the motor end plate. The depolarization will travel down the muscle cell and into the T-tubule pictured in diagram A. This causes a depolarization of the voltage-sensitive dihydropyridine receptors, which is also coupled to the ryanodine receptor on the intracellular sarcoplasmic reticulum. This leads to a conformational change that causes calcium to be released from the sarcoplasmic reticulum. That quite literally means calcium was released from the reticulum because calcium entered from the outside. The high level of calcium binds to spots on troponin C, think C for calcium, which then causes a conformational change, causing tropomyosin to move out of the myosin binding groove on actin. You can remember that tropomyosin is the adapting molecule between the calcium-binding troponin and the contraction-generating myosin portion by keeping in mind the name incorporates both sides. Myosin then releases prior-bound ADP molecules and is displaced on the actin filament, the so-called power stroke, which is actually how myosin molecules move along actin. We'll look at this concept more in detail on the next page. Do you know why bodies develop rigor mortis after death? It's because after death, ATP generation ceases and there's no more ATP available to bind the myosin and release cross bridges. Before we do that though, we're going to discuss different types of muscle fibers. It might seem a little tricky at first, but it's actually just a quick fact to tuck away. Basically there are two types, type 1 and 2. You should think about type 1 as being responsible for sustained contraction and 2 for being responsible for fast twitch. Type 1 fibers are slow twitch, and you can remember everything you need to know about them with the memory aid one slow red ox. That is to say, they're slow twitch, they're red fibers, meaning they have a lot of mitochondria and myoglobin, and ox, which means they utilize full oxidative phosphorylation in their mitochondria. Conversely, which metabolic pathway, glycolysis or oxidative phosphorylation, will the white fibers rely more on? Exactly, glycolysis. As you'll remember from the biochem chapter, it's much less efficient and requires a significantly higher amount of glucose input per ATP versus oxphos. Remember, think one slow red ox. One good example of slow twitch muscles are the paraspinal muscles, which have to work consistently for long periods of time to hold the body in position. Slow twitch muscles tend to be postural muscles, for example your paraspinal muscles. They have long sustained contractions. Just think about when you're walking or sitting up. This allows ample mitochondria to have time to go through oxidative phosphorylation and create enough ATP to continually power these muscles. In contrast, fast twitch muscles are white because they do not have a lot of mitochondria or myoglobin. They primarily use anaerobic glycolysis. If you think about it, that's probably for the best. Fast twitch muscles include muscles like your biceps or your extraocular muscles. They can't wait for additional ATP to be generated through oxphos. They need it fast. This muscle is also the type of muscle that commonly hypertrophies in weight training. As promised, we've now returned to the topic of muscle contraction. Before we were looking at the sarcomere and its greater organizational structure within muscle fibers and even muscle fascicles. Here we'll look within the sarcomere to see why the areas of actin-myosin overlap that we discussed earlier actually contract. The basic idea is that heads from the myosin chain attach to actin and stroke along them, a process that's dependent on ATP. You can think about the first step as just having a myosin head that's ready to attach to actin. In the resting state, that myosin head can't attach because tropomyosin blocks binding. However, when the signal for muscle contraction comes, calcium rushes into the cell and binds to troponin C, causing the tropomyosin to move out of the way and allow binding of actin to myosin. The myosin is presumably already in the cock position, so the next step is for the myosin head to power stroke along the actin, resulting in the loss of a phosphate group. Subsequently, ADP falls off the myosin head as well. This is what triggers the power stroke, causing the actin and myosin to slide past one another and causing contraction on the larger scale. Soon, an ATP molecule will rebind the myosin and cause it to fall off the actin. Think of the T in ATP as standing for terminate, as in it terminates the connection between actin and myosin. The energy from the ATP is then used to reset the myosin head into the cock position, leaving behind an ADP molecule in the myosin head. Repetitions of this cycle lead to repeated cross-bridge formation and shortening of the sarcomere. Now we've talked a considerable amount about the physiology of muscle, which admittedly is pretty complicated, but before we turn our attention to the path section, we've got one more topic, and that's bone formation. There are two processes of bone formation, endochondral ossification and membranous ossification. The difference between the two is pretty straightforward. With endochondral ossification, cartilage is laid down as a precursor for bone growth. It's sort of like making an outline for an essay. After making the outline, you have a structure already that you can follow for writing the first draft. After you make the first draft, you can go over it one last time and make a final draft. That's basically how endochondral ossification works. You start with a cartilage model, or the outline, and then convert it into woven bone, or a first draft. While woven bone is actually bone, its fibers are haphazard and lack strength. Woven bone is then converted into lamellar bone, or the final draft, which is more structurally and mechanically sound. This is the method of formation for all long bones in the body, including the femur. Extending the metaphor, it takes a lot of drafts to make a long essay, just like it takes a lot of formational steps to make long bones. The second process, membranous ossification, isn't entirely dissimilar. It's basically writing the first draft without the outline. There's no cartilage, there's only a first draft written from scratch, or woven bone, laid down without a cartilage framework. Woven bone is then refined into lamellar bone, or a final draft. The process isn't used for long bones, but rather for the flat bones. Can you name a few flat bones? The skull and facial bones are some good examples, which as a side note are the major sites for active erythrocyte-producing red marrow later in life. Now that we've talked a little bit about bone formation, let's bone up on the different unique cells they're made of. Osteoblasts are builders. They secrete collagen, the protein matrix around which the mineral portion of the bone mineralizes, and also help catalyze the process. They're derived from mesenchymal stem cells in the periosteum, so if the periosteum gets damaged, they have a harder time regenerating. Osteoclasts, which have a C, like the word consume, generate HCl and collagenase to break down and resorb bone. They differentiate from monocytes, just like macrophages. The main thing to keep in mind about PTH is that low intermittent levels lead to buildup of bone, whereas chronically high levels lead to breakdown. Estrogen is also important in bone homeostasis in that it prevents apoptosis of osteoblasts and induces apoptosis in osteoclasts. When estrogen is lacking, either due to menopause or surgery, osteoporosis can result.